Great, thank you very much. I'm a professional research scientist, so you get the professional research scientist who's got an outreach team behind him. Opening slide. It doesn't need much explanation except remember the logo. And there's two Twitters because the first one is the boring work one, and the second one is where I talk about Brexit and the local cricket team. So after the talk, choose which one, if either, you'd like to ever look at. And to tell you a bit, in order to tell you about what I've been asked to tell you about, I need to tell you about the place where I work, and this is the European Bioinformatics Institute, which is a multinational, multi-government funded, not-for-profit research institute just south of Cambridge, where it is always sunny, like in the picture. You may or may not know, but genetics and molecular biology is a fairly big data kind of place to work these days. We are one of the, it says a major data provider, but it doesn't say the whole thing. I mean, we are probably the leading world repository for the kind of genomics and genome data that hundreds of thousands of scientists and medical research centers and so on are generating. So we, these days, have a data center that runs to over 150 petabytes. We have 38 million requests for our computers to do something with that data and give it to people per day, from over 3 million different locations a month. So this is fairly big data, and I'm not going to give the big data talk to this crowd. But we have graphs where things go up. So if I walk in this direction, time is passing. And as you go up, the size of the various databases where we store large amounts of this information for the scientific community goes up. And the lines, if you can see the lines are, if it doubled every year, you would be going along this line. So it's a logarithmic scale, so those are straight lines. So actually most things are not quite doubling every year, but then sometimes a new technology comes along and you get a very scary jump. And we are a bit like, it's been described as like it's an organization which has 60 10-person startups inside it, each one of which is handling large amounts of data, developing web services that interconnect, that talk to the whole world, because there's a large world. The world for us is the scientific community that want to share data, and it's a very open community, an exception if you're working inside a hospital. There's an awful lot of data sharing going on. And the little picture on the left is that the little bits of writing you can't read are some of the major data repositories that our institute looks after for the world, and the complicated spaghetti thing is the way they interconnect. And these things go up. The blue curve on the right-hand side is the kind of raw DNA, genome data that we're asked to store and serve back to the entire world community. And as it started taking some of these jumps in the past, we got very worried about our funding does not go up logarithmically or exponentially, and we got worried about whether we would be able to store all this data for everyone in the whole world. And I was at a meeting a few years ago where we were, after the actual meeting where we worried about it, we were in the bar afterwards, and we would say, well, if only there was some other way of storing it, because of course we're using hard disk drives and data centers and a lot of air conditioning, and it's all very expensive. Isn't there some other way of doing it? And we're looking at graphs like this saying there's some other way of storing information. We realized that, the blue one is the amount of information we are storing about genomes, and genomes are just information, and they are stored somewhere, and that is in the DNA of every cell. Actually, I was going to say every person's genome, but I mean every cell, which is tiny, has all that information in it. And we have this thing called DNA, which can store information. And then we wondered, well, maybe this would be a useful thing we could do for real, and one day overcome the problems of data centers expanding to cover the surface of the Earth and so on. So everyone's heard of DNA. How many people kind of know what the diagram is? Quite a lot, okay. Well, for those that don't, and because it helps my flow keep going, DNA is a chemical, And it forms a chain of 4 different molecules with the names on the right. A, C, G, and T is the letters used to represent each one. And in your genome, you have a number of pieces of DNA, each one many millions of bases or letters long. And that stores all the information that's needed for a cell to build another cell, to do all the processes that a cell needs to do, to metabolize nutrients, to reproduce, to split into daughter cells. And that's a fantastic information store. And it's really, really small. These are numbers that I don't really know what they mean. I can't imagine them. I know what they mean. But each link in the chain is 4 angstroms, or less than 10 to the minus 9 meters long. And so this diagram shows it in the famous double helix representation, where you've got essentially 2 copies of the same information, but using a different code on the different strands, and they will stick together. Essentially, there's a chemical bond between them, which is incredibly stable, and it means you've got this system where if you can split those up, then each serves as a template for making the other one, and this is how DNA genomes are copied in living cells. And I pressed the wrong button. There we go. But the information is actually on one strand or the other strand, because once you know one, you know what the other one is. There's a set of rules about how it matches up. And in fact, the information, you can just think of it as a series of the letters A, C, G, and T. So that's where information is stored in genomes, and we wondered if we could use that. I mean, we're used to seeing things like this, but it would typically be zeros and ones, and everyone in this room would clearly be very comfortable with the idea that we represent information in a string of zeros and ones, and that our computers know how to write that, and they know how to read that, and they know how to interpret that, and that's what all computing is based on. Except maybe quantum computing, I suppose. I'll have to update the talk soon. So we wondered, maybe now we've stumbled across this fantastic new solution to rid the world of huge data centers. Can we do this for real? And what you need to do, to transmit information is you need a system for writing it, and you need a system for reading it, and you need a system for having, a language or a code to put the information in. And we realized that we actually can just about do all those things on a useful scale these days. And these are all innovations driven by the genomics revolution and the fact that we want to sequence 1 human genome, and then we want to sequence 1,000 human genomes, and there's currently 100,000 genomes projects. And that's before you even get into cancer, where you need to sequence every single tumor separately to understand what's going on. There's an enormous amount of genome work going on. The first thing here is the writer, and humans aren't as good at writing DNA as we've become at reading DNA, but we can do it. These pictures, the main pictures to the left-hand side, from a company called Agilent in California, and they have a system. It essentially needs a clean room, and it has a machine based on an inkjet printer. In fact, they're a spin-off from Hewlett-Packard. And they're using what's essentially an inkjet printer to deliver the chemicals that make up DNA very accurately to different locations on a glass slide, and they can build up these chain molecules kind of on demand. We can't make them very long yet. They can do a couple of hundred bases, letters long in each chain, unlike your genome, which would be millions or tens of millions long in each chain. But we can make them. It's really valuable for doing biomedical research, so there is still market forces are driving this forward, and the little pictures on the white backgrounds at the bottom are just some of the publicity shots from a company called Twist Biosciences, who are a startup that are doing the whole thing, but smaller, faster, cheaper, by having even more accurate control of the way they do it. So we have a DNA writer. It's not very convenient yet, but we have it. And we have a reader. The big picture down on your right is what you used to use about 5 or 6 years ago if you were sequencing genomes. And it was a machine that size. You can see my colleague Paul Bertoni just slightly cut out of the picture because I'm not a very good photographer. And that's the machine you need to sequence DNA, to read DNA. And under the table there's a computer, and all the information is written on the computer. And that's typically what's used if you have your genome or any living cell's genome read. That was a few years ago. That the current kind of new kid on the block is a company called Oxford Nanopore. They're making this device, which you hold in your hand. That does essentially the same as this one. It's a bit more error-prone. You can't get quite as much read on that machine as you can on that one in a given amount of time, but it's just that device. It's easily portable. People take them out all over the world. It plugs into a laptop computer. And you're off, and you're sequencing. So there's our reader. The genomics revolution has driven the development of these, and in the next few years, this one will have gone, but there will be another one. And you need a code, you need a language to write information in. So part of my research life is studying the way evolution affects the information in our genomes, and this is the very famous genetic code discovered in the years after Watson and Crick discovered what DNA was. And that's how groups of 3 letters, A, G, T, whatever it is, encode the different proteins that make up a living body. But you don't need to know this. There won't be a test on this. The important point I need to make here is that what this doesn't do is tell us how to encode PDFs or MP3s or whatever it is we want on our computer. Evolution has designed this to encode the instructions for making a protein, but not much else. And so really the only contribution I made in this study, in this research, was to invent a code. You know, the reader was already there, the writer was already there, and I needed to just have a way of representing information in DNA. And it's really not that difficult. You're just going from a 2-character code of 0s and 1s, like represented at the top, through some intermediate steps, but so that by the time you get here, you're representing it in a 4-letter code, A, C, G, and T. It's actually trivially simple, except we know there will be errors in these processes. The error rate is much higher than with a hard disk drive and so on, both in the creation of DNA, the synthesis of DNA, but also in the reading. We have a reasonably high error rate, but it's not terrifyingly high, and you just need some kind of error-correcting techniques, and we did some absolutely simple ones designed on reducing the number of errors. So, for example, we never allowed the same letter to be repeated. The code, although it has 4 letters, never has any repeats. That's easy to design in. It helps us out a little bit. We also did some very obvious kind of redundancy by making a design and then making fragments of DNA which overlapped. And then, unfortunately, when you write them and read them, you don't get them back in any particular order. It's like, you know, DNA comes in a test tube in liquid, and when you read it, you fish out pieces in a random order. So we needed to add a kind of indexing procedure as well. But basically, it's all very straightforward and used my undergraduate maths degree coding systems. And this gave us enough ideas to devise this experiment, which is you start with some information on my laptop computer. You upload that or you put it on a web page. Agilent in California can download the information. They put it into their inkjet printer type machine. They make actual DNA according to the designs that we've come up with that could carry that information. They send it to us by FedEx, actually, but this is suitable for the BBC version, which just says courier. They send that to me back in Cambridge. A bit of the lab work gets done in Cambridge, but actually at that time, the DNA sequencing machines that my institute had access to were in Germany, so that gets sent to the sequencing machine in Germany. That writes the information on the disk. File transfer rates are too slow still in 2012 to do this, so my postdoc researcher comes back from Germany with a hard disk in his pocket. We plug it into my laptop, we do the decoding, and we look to see whether we've got the right thing. So that's the plan. What did we store? Well, things that will get us some good publicity, basically, because we thought we were trying to get this published in an exciting journal. So obviously, one of the most famous journals is the journal Nature, and that's the one that in 1953 published Watson and Crick's description of the structure of DNA. So we went and got permission from Nature and said, please may we have the PDF of the publication. I mean, obviously it wasn't made in 53, but they've subsequently created a PDF of that. Please may we have that, and can we write it into DNA for this cool project we're doing? And please will you publish our paper when we finished? So that went very well. But of course, it's not just we needed to, for people that aren't savvy about the way computers work and don't think that a PDF is essentially the same kind of information as a JPEG or a text file or whatever, we thought we'll use a variety of different file types, and that will really impress people. So we picked all of Shakespeare's sonnets, because it sounds like a lot of information, but it's not really very much. And the famous photograph you saw before, which in fact I took from on the site where we work, and my office is this one, if you want to know, and my collaborator Ewan Burney is the office above nowadays. And so it doesn't matter because they're all zeros and ones, right? But it's text, it's JPEGs, it's a PDF, which is really just a JPEG inside some kind of wrapper, I think. And that's a picture of Martin Luther King, but we didn't use a picture of Martin Luther King. We used an excerpt from the actual recording of him giving his I Have a Dream speech, because we thought, oh, we do sound as well, and that will really impress people. And of course, we would have done video, but the file size is getting a bit big for our kind of proof of principle experiment, so we didn't do video. So we picked those, and we did this experiment, and we got it all back, and then we said, well, did that work? And this doesn't make for a great, well, I'm getting ahead of myself. We did the experiment. They sent me the DNA. It comes in a box like that. It comes in little test tubes, or Eppendorf they're called. They're about as big as 2 joints of your little finger, and you look at them, and it's empty. And you think, oh, this has gone wrong. I don't understand. I'm only a mathematician. What's gone wrong with the DNA? And you go down the corridor to where your colleague Paul Bertoni works, and because he knows what he's doing in a laboratory. And he said he teaches you that what you have to do if you want to see the DNA, if it's been dried. So DNA is essentially a powder if it's dry. When you see a little drop of liquid, it's because it's been put in solution. And when it's dry, which is how they sent it, there's essentially nothing visible to see, and he teaches you how you hold it up to the light. This is me holding it up to the light and pointing at it, because you can just about see a kind of smudge of dirt in the bottom of the test tube. And I think the good description, if you imagine you had some salty water in a test tube, a tiny bit, a drop of salty water, and then you let the water evaporate away, you kind of get a film of salt just kind of visible on the edge. It looks like that. I mean, DNA is really unthinkably small. So that arrived, and it went through the laboratory in Cambridge, and then it went, you've seen this picture before, this is our sequencing machine, this is Paul, this is taking place in Germany. It's one time I was allowed in a lab, but they didn't let me touch anything. And you do the whole decoding thing, and all I can say is I can either show you a load of bits and show you they're the same, or ACs and Gs and show you the same, or 2 copies of the same picture and say they were saying 2 copies of the same little bit from a PDF. But the point was we got every single bit back correctly. So we'd encoded nearly a megabyte of information into DNA, gone through the whole process of creating the DNA, handling the DNA, reading it back, and we were able to put every single bit back in the right place. And I got a couple of minutes. When we did this, we got a bit of publicity, and a Swiss journalist phoned me up and said, I saw that you'd published this and you got the picture. Could I have a copy of the picture for our magazine? And I said, well, it's all online. And she said, well, no, but the one you put online is the one before you did the encoding, and I want the one after you decoded it, after DNA. And I said, but it's the same. And she said, no, I don't, I not just said it looks the same. And I said, really, it is the same. I didn't just look at them. I actually, got the computer to, so, she wouldn't take no for an answer. So I emailed her a copy of the photo, and to be honest, I don't know which copy it was. But she phoned back the next day and was delighted because they were the same, apparently. So that was good news, a weight off my mind. So we did that a few years ago, and that was great fun, and we, I call it Friday afternoon project, because you kind of do it a little bit at a time when you've got a bit of spare time, and I'm a big fan of having that kind of project to do. And we did, Nature kindly published our paper, so that's kind of a win for the scientist in me. And it got quite a lot of publicity, and there's still people talking about it, so I do a quick what has happened since, because it's not just us. We did some of the first work on this. These are some of the people we've spoken to in the last few years who are taking an interest in this, and they have various reasons, but it is, to do with the fact that people see a problem with the way we're currently storing data, and it can't necessarily go on the way it is. And also, I have a whole different talk on this, but we're really, at the moment, there is no archiving of digital information going on anywhere in the world. And by archiving, I mean you put something somewhere safe, and then you just leave it there, and it's safe. So we got the, in the UK, in London, we have the British Library. You put a book in the British Library, and basically it's safe, and it's there forever. There's no equivalent for digital information because someone is actively managing a storage facility of some sort and replacing hardware and spending a lot of money doing that. They may or may not ask you for that money if they're funding it by advertising or whatever, but they're doing it. Technicolor is very worried. The film industry has noticed that they have got assets that are now coming on for 100 years old that are currently stored on celluloid, and that's not working out very well. And then the new ones are coming digitally, but they know that's not a good backup store. So the archive copy of the latest digital movie is often now done they put it onto celluloid because they know they can keep that for 100 years without touching it, whereas they know if they leave it on a hard disk, it'll be dead in 5 years. So they're the ones that came to us and said, we're really interested in new information storage technologies that do not involve spinning rust. So that's a good system. But Microsoft, if you go and Google this kind of thing afterwards, you'll see Microsoft are investing tens of millions of dollars in a program to investigate whether DNA will be a viable information storage technology in the future. Huawei have just come out from behind the clouds or wherever they hide and have said the same, but we probably won't be allowed to use their solution if they do get one working. IBM, interestingly, I had 1 week a couple of years ago when I, within 3 days, spoke to one person at IBM that described themselves as the lead on their DNA information project, and then the next couple of days spoke to someone that denied they had a project looking at DNA. So who knows? One of them is deluded or lying. And then most recently, these are some really just some nice pictures, but this is the University of Washington who are working with the Microsoft team, and they've made now a device. I showed you pictures of machines. That's one device. It's a bit ad hoc, obviously, but it now fits on one table, and at this end, it creates DNA, so it gets a message from a computer. It makes DNA. It stores the DNA, it dries it out just to show that that's possible. It adds liquid again, and this is actually a version of one of those handheld devices. It's got the lid off, so it doesn't look exactly the same. But they've got the whole thing now working on the table, on one table. Obviously, we want the whole thing to be something the size of a memory stick, and hopefully that's coming. And this is a lovely picture from a collaborator of mine, Robert Grass, and his student Julian Koch in Zurich. And they're thinking about ways that we can take DNA that will have information in it and keep it safer and use it for things. And they've got a system where they get DNA and they kind of trap it inside a glass bead. And once it's in there, it's much safer, it will last, it's quite resistant to heat, it's totally resistant to liquid or wet, which is one of the big problems with DNA. So each dot is a little glass nanoparticle. And each one of those has something like 17,000 bits worth of information entrapped. So roughly speaking, most of what you see is glass, but in there's 17,000 bits of DNA in each one. And what they're resting on is the surface of a DVD. And this is a scanning electron micrograph of the surface of a DVD. So this, where's it gone? Each little mark is 1 bit in DVD world, or tiny spot like that is 17,000 bits if you do it in DNA. And that's not really DNA. You're really seeing the glass rather than the DNA. So people are, we're pursuing this. We're trying to get there. We'll see if we can have a working product in a few years. But why are we doing this? Well, I've explained it's really small. Why are we not doing it? It's really, really expensive, and particularly it's the synthesis, the writing of DNA. We're not there yet with that one. We need a few orders of magnitude improvement, which sounds scary, but then those of us that have been working in genomics are not worried by, we've had 6 orders of magnitude improvement in reading DNA in the last 10 years or so. So if we can do the same with writing, we're going to be there. If you can write it at the moment, you can afford to read it. In fact, the reading speeds and costs are not as good as we'd need. But we know they're improving. They've been improving for a long, long time at exponential rates. So that all looks good. But we would do it because, it's really, really small. So I told you, something you can't see stored about 1 megabyte. If you'd filled that test tube up, so 2 joints of your fingers worth of DNA, it's roughly equivalent to 1 million CDs, and my demo of 1 million CDs is I work out the size of the stage I'm on, which I have done, and so this is something in the order of 30 square meters. So let me get this right, 10.3, it's about It's about 3 and a bit stacks of CD-ROMs. So if you imagine the whole stage, was filled with CD-ROMs about up to my knee, and I was wading around in that, that's the equivalent amount of information as 2 joints of your finger, test tube of DNA. So it's really dense, and the sciencey bit says the indexing system we use does scale up to monumentally huge amounts of data, but you can ignore that and you can go home thinking, that guy said you can get all the data in the whole world in the back of a van. And currently you can. I think that's still true. I did the calculation a few years ago. You might need a slightly bigger van at the moment, but it's that kind of order of magnitude. DNA is interesting because it's stable. It lasts a really, really long time. And my evolution studying part of my work life teaches me this because we routinely have DNA samples from things that are thousands of years old. And these are some of the classic examples of the older ones. And the best one around still is this ancient horses, where they were reading genomes that were over 500,000 years old. They weren't carefully prepared scientific samples. They're just stuff that died somewhere cold. And that's because what you do need for DNA to last is that it's cold and dry, particularly that it's dry. And that's easy if you're a global data provider. You set up something like the Svalbard Global Seed Vault, which is an international collaboration that keeps seeds cold and dry by sticking them in a hillside in the Arctic Circle. In a sense, they're just storing DNA, right? But they're doing it in seeds. And we would want to do it in test tubes. And if you don't have the facilities for that, you probably do have the facilities for keeping your DNA safe at home. One of those, particularly the top section, will keep it safe for hundreds, if not thousands of years. The fridge would die before the DNA. I didn't really mention it before, but DNA is fantastic for making copies because the message in the DNA is, in a sense, the machine that makes the copies. Because humans have learnt to reproduce the process that goes on in cells of splitting the DNA using each as a template, then you multiplicatively copy it. So if you want 1,000,000 times as many copies as you've currently got, it takes about half an hour in the laboratory. Unlike the process for copying paper documents or making hard disk drives, which is a very linear process involving a lot of complex machinery. And the last reason is that you always have to check the age of the audience, and over the years I become the oldest person in the room. I've used all but 2 of these technologies, you can guess which ones, but I don't use them anymore because they are obsolete, because the medium that stores the information does not survive that well, and if it did, we haven't got the machine anymore to do the reading. This is kind of a selection of the things we use nowadays. How many of these will still work in 10 years? The kind of timescales we want to store some information, or 50 years, or think of the British Library, hundreds, many hundreds of years. Probably none of them. But DNA will always be there because as long as humans are interested in their own health or in studying the biodiversity and the planet, we will be reading DNA. The machine we use to do it will change, but the medium remains the same, and there will always be a machine. So I think DNA's got quite a good future. That's all I really wanted to say, but I noticed it was okay for speakers to do a quick plug of their own interests. We don't often get out to these kind of developer meetings, but I'm not hidden at all. The vital part of what goes on in our institute is done by over 300 developers who don't always know we're there and don't think we're the first place to go for when they're looking for a job. But we are essentially always recruiting. There's a list of technologies, the words of which mean little to me. But I think a lot to you, and I think they were chosen because they appear in the program of this meeting. So if by any chance anyone has come here because they might be looking for another job in the I know it's unlikely, but if that was happening, I'll be around for some of the rest of the day. My colleague, Jonathan Hickford, is in the room as well and has a matching T-shirt, so you can find us. So that's the little plug, but I'll get off because I know I've overstayed my welcome slightly anyway. So I'll go back to this one so it's not too blatant a plug. But thank you very much for listening.